My name is Michelle Dion, and I'm a PhD candidate in the Medical Engineering and Medical Physics program at MIT and Harvard Medical School. And today, I'll talk to you about our work on the development of a tunable tissue-adhesive biomaterial for the treatment of glioblastoma. Before I begin, I just wanted to quickly acknowledge the support of my lab mates who helped in this research work, as well as our collaborators in Johns Hopkins in neurosurgery, Dr. Henry Brem and Professor Betty Tyler, as well as my support from my advisor, Natalie Artsy, and the continued support of the Deshpande Center, particularly our catalysts, Jason and Christine. So as many of you know, innovation and the development of technologies does not happen in the bubble, and it does not happen overnight. So as you can see here, the development of our adhesive, tunable delivery technology has happened over time. It's been spanning multiple therapy types, multiple tissue types, and we have most recently innovated on it to bring it to brain cancer therapy for the delivery of chemoimmunotherapy. But why brain cancer? So at MIT, we like challenges, and we like to meet an unmet need. So in glioblastoma, over 80% of patients will not see their second year of life after diagnosis. This is one of the most aggressive and the most lethal of all cancers. And this is even with current therapy, which consists of surgical resection, radiation, and chemotherapy. And even new innovator therapies, such as immunotherapy, have not been able to budge this survival rate. So why is that? Let's look at a clinical scenario. So as I said, about 80% of patients will receive surgical resection to remove the tumor burden and to extend survival, as well as to reduce the effects of the tumor's presence on the side effects the patient experiences. As you can appreciate, surgical resection is not able to remove all tumor cells, so patients will begin to undergo chemotherapy systemically, as well as radiation therapy. However, with this aggressive and heterogeneous tumor, they aren't able to eliminate all cells, and eventually the tumor recurs and infiltrates to different regions of the brain, including the contralateral hemisphere, or the opposite side of the brain. So again, why do these therapies fail? As our clinical collaborator at Dana-Farber, David Reardon, said, the top 3 reasons that brain cancer therapies fail are because of 1, drug delivery, 2, drug delivery, and 3, drug delivery. Now, as a drug delivery scientist, of course I'm going to agree with this, but it also has a really important biological reason as well. So as you can imagine, the brain is a vital organ, and as such, it is well protected. In this case, the blood-brain barrier prevents over 98% of systemically administered therapies from reaching the brain. This is an evolutionary advantage because, of course, you don't want things like COVID-19 or bacteria to be able to reach your brain easily. But you also want things like nutrients and oxygen to be able to get into it. However, in the cases of things like chemotherapy, which looks quite like toxic agents, the blood-brain barrier isn't able to distinguish between a helpful therapeutic and a toxin. And this is even in the case when you attach it to a nanoparticle or an antibody-based conjugate. This is particularly important when you think of cancer like glioblastoma, which is heterogeneous, which means it has different types of tumor cells, which can be responsive to different therapies, which means that different therapies may not work well for all cell types, suggesting the need for combination therapies. And even when you can get therapies across the blood-brain barrier, they may not be able to reside in the brain at therapeutic levels for therapeutic time periods. To solve this challenge, we've innovated on our adhesive biomaterial to be able to inject, spray, or paint it locally into the tumor resection cavity or into the tumor bed directly. This enables the controlled release of therapeutic agents. One of the things that distinguishes our hydrogel-based technology is that it is adhesive. This means that the chemical reaction that governs hydrogel formation also governs the adhesive interactions between the materials and the tissue. This keeps it in place, as well as reduces the diffusion distance of the drug to the tissue. Furthermore, as I'll show, we are able to tune the degradation kinetics of the hydrogel such that it can release therapies at controlled rates, and that it is also able to be removed from the brain. Furthermore, we've shown that it is compatible in multiple small animal as well as larger animal models in systemic organs, as well that it is tolerable. And finally, as I mentioned, combination therapy is important with glioblastoma. And here we'll show that we're able to achieve therapeutic outcomes specifically with this local-based administration. But let's get to my favorite part, the data. So, starting with a hydrogel that is the size of a raindrop implanted by injection into the mouse brain, we were able to follow with orthogonal methods for both fluorescence imaging in vivo as well as MR imaging in the brains of healthy mice. And you can see from the two curves that the degradation kinetics of the hydrogel is both controlled by the material formulation and can be continued over the course of 3 to 4 weeks in the mouse brain. We have further showed that the payloads that are released from the hydrogel remain in the brain, and even when they are cleared systemically, do not remain there in systemic levels above background noise, as shown in the graph on the right. With these pharmacokinetic profiles, we were excited to move this technology into mice. So here you can see what current oral chemotherapy looks like in a mouse model of glioblastoma. And you can appreciate that even with current therapy, mice don't survive much past the 26-day mark or 1 month mark. Even when we overcome the blood-brain barrier by administering the doxorubicin therapy locally, we see that this doesn't significantly extend survival. However, when we use our hydrogel-based drug conjugate to locally and sustainably release the therapy, we were able to get over 30% long-term mouse survival. And when we powerfully combine that with an in situ vaccination approach with an immunotherapy adjuvant, we were able to get over 80% of mouse survival in this model. Excited by these results, we further re-challenged the mice with an additional tumor. This means that we put it in the contralateral hemisphere and gave the mouse no more treatment. And when we studied their survival for these mice, we saw with the in situ vaccination approach that we were able to get 100% of long-term mouse survival in this mouse model. And we've seen similar efficacy in a secondary mouse model as well of glioblastoma. Inspired by these results, we've recently spun out our company into Arrhenius Bio. We have experienced leadership in both oncology and material platform development, as well as being advised by some of the top neurosurgeons in the world. Many of our team members are here today, and we'd be happy to speak to you more, because as with everyone, we have a few asks. The first, like everyone in the biotech space, is we're obviously looking to be connected with potential financing opportunity, as well as partnership opportunities, both at the CMC level for manufacturing the drug conjugates, as well as the therapeutic level for identifying new agents that could benefit from our technology. Because we know that it will take all of us to be able to deliver efficacious results to patients who need it most, and that we believe we can do this by eliminating cancer at its source. Thank you.